CRISPR could be responsible for a new era of genetically perfected plants, animals, and even humans, but what the heck is it?
Revolutionary technology that can edit genetic mistakes is getting attention and scrutiny this morning.
It's been called the scientific breakthrough of the year.
The CRISPR technology is like software for the genome. We can program it easily using these little bits of RNA.
A few years ago, something called clustered regularly interspaced short palindromic repeats, or CRISPR, burst into the scene. It worked so well, scientists began issuing ethical statements about its use. CRISPR cuts DNA strands with unprecedented accuracy and simplicity, allowing geneticists to directly edit any of Earth's organisms however they like. CRISPR could be used to engineer disease-free organisms, formulate high-yield crops, or even cure genetic and hereditary human conditions. Of course, it could theoretically also be used to let parents pick their kids' sex, eye color, height, or whatever. In the end, CRISPR is, as one Nobel scientist said to The Independent, jaw-dropping.
It lets scientists rearrange the building blocks of life, removing one gene, adding another, manipulating DNA with unprecedented ease and precision. And the CRISPR excitement is building around the world.
Meeting in Rome, and there was a presentation, and the guy just knocked our socks off. It was just, all of a sudden you're just sitting there and going, oh my goodness, what I could do with that.
So I'd like to show you my lab.
What Elizabeth Simpson is trying to do is cure genetic blindness in children. And to do that, the Vancouver geneticist has built the gene mutation into mice.
So that is a sign of...
The mice have been engineered to develop the same disease as a child with the same genetic mutation.
Okay. So that's a normal.
Yeah, exactly. So that's what a normal mouse eye would look like. All right, so if I put her back and I see if I can find her sister that has a mutation. Okay, so here we go.
Oh,
so you can see right away that the eye itself is quite a bit smaller. And if we get a really good close-up shot, you'll actually be able to start seeing in the light that there's a whiteness over some of the eyes, particularly on this right eye here.
So is that mouse blind?
It's not completely blind, but it's much closer. It's very vision reduced. And our hope is that by correcting the genetic defect that these mice are born with, that we can actually restore the normal eye shape and function.
If they can reverse the blindness in the mouse, in theory they should be able to do the same thing in a child. So far, it's worked.
You have to also consider that the CRISPR technology can be used for things like enhancement. Imagine that we could try to engineer humans that have enhanced properties such as stronger bones or less susceptibility to cardiovascular disease, or even to have properties that we would consider maybe to be desirable, like a different eye color or to be taller, things like that. Designer humans, if you will. Genome-engineered humans are not with us yet, but this is no longer science fiction. Genome-engineered animals and plants are happening right now.
Important is CRISPR. I think it's the most important thing I've ever encountered in my scientific and clinical career.
At Sick Kids Hospital in Toronto, Dr. Ronald Cohn believes CRISPR might hold the key to repairing genetic diseases.
So then there's a good chance that we see a duplication. I became mesmerized by this technology and by the potential options. I pulled my lab together. Very exciting. That was now almost 2 years ago when I changed completely the direction of what my lab has been focusing on.
So CRISPR changed your research completely?
Completely.
Right now, Dr. Cohn and his research team are working with the cells of a 13-year-old boy named Gabriel. He has Duchenne muscular dystrophy, a genetic mutation that has already cost him the use of his legs and will eventually leave him unable to move. So far, Dr. Cohn has used CRISPR to fix the genetic mutation in Gabriel's cells in a petri dish.
About people who will hear about this technology, who have the financial means to tell the company, said, can you please create a CRISPR drug and make me more muscle, make me smarter, make my eyes green. Eventually it's all going to be technically feasible, and I think that's why we need to put a bit of a framework here in place so this doesn't get out of control.
Meaning scientists could cut single genes out of a DNA strand. After the strand was cut, it would self-repair, disabling the gene. But if scientists injected replacement DNA, that would fill the space instead. This allows us to swap in DNA wherever we like. You want corn with genes to fight bacteria? Cool, can do that. Want fish that glow? Grab some DNA from phosphorescent algae, toss it in there. This technique is so simple, it's scary. It's paving the way for widespread genetic engineering, and it's kicked off a media frenzy. Now, CRISPR-Cas9 isn't perfect. Sometimes the process will target the wrong section of DNA and cut there. But a new paper in Nature describes how scientists altered the Cas9 enzyme, reducing off-target mutations to undetectable levels. So they improved on evolution, and now it is perfect, and even more scary.